Hello, and welcome to this video on the mechanism of DNA replication. At the end of this video, you will be able to describe the features of DNA polymerase, describe the evidence that DNA replication is semi-conservative and semi-discontinuous, explain the mechanism of DNA replication, and explain the mechanism of telomerase. To start, remember that DNA is a template for its own replication. So one of the things that we know is true is that DNA is read 3' to 5', and built 5' to 3'. So in this picture, if we had the parent strand over here, and it's anti-parallel and it's double-stranded, during replication, one of the things that we will see is that these hydrogen bonds will be broken, and each of those strands will become a new parent for two new daughter strands to be built. So if we were to replicate this parent strand, there would be a DNA polymerase at the 3' end of the top strand, moving to the left as drawn, and a polymerase at the 3' end of the bottom strand, moving to the right as drawn. And they would build those new daughter strands, moving off in either direction, so that the double helices that result are identical to the original parent DNA double helix. There are 4 features of DNA replication. The first is that DNA replication is semi-conservative. And what that means is that each new DNA double helix includes one of the original parent strands and one newly synthesized strand. This experiment by Messelson and Stahl is what showed that it's semi-conservative. In this experiment, Messelson and Stahl started with DNA that they grew in what's called heavy nitrogen. So all of the nitrogens here are N15. And they did that for many, many generations. And then they collected those cells and they washed them and they grew them in a new medium of N14. And they looked at the result. And there are 3 possibilities here. One possibility is that they got only N15 or N14 pieces of DNA. This is the conservative model. Another possibility is that each resulting piece of DNA is some combination of N15 and N14, but that it's very difficult to predict which pieces came from the original parent and which are new. We call this the dispersive model. The third model suggests that each parent strand, which in this case is N15, is used to create one new daughter strand, which in this case is N14. And we call this semi-conservative. What's so great about this experiment is that they did it in bacteria. And the great thing about bacteria is that they double their genomes and they divide every 30 minutes. So in a very short amount of time, we could see whether the resulting daughter strands of DNA are the original parent strands, N15, or have been newly synthesized and are N14. The way that they do that is by putting the DNA onto what's called a cesium chloride gradient. And then they spin it in a centrifuge very quickly, and the DNA distributes down this gradient, and how far it goes down the column depends on how dense the DNA is. So we see in this picture that after 0 rounds of new replication, all of the daughter strands are heavy. They're N15. After 2 rounds of replication, we see that there's a mix of N15 and N14 strands. And in between, when we have only done 1 generation of replication, after about 30 minutes, we see something that is in between N15 and N14, indicating that it is probably a hybrid. So after the first round of replication, we have only hybrids. But after the second round of replication, we have some daughter DNA that is N14 only, which is exactly what you'd predict from a semi-conservative model. A dispersive model would always have hybrids. The conservative model would always have some N14 only. Only the semi-conservative model is represented by the data here, where after 2 rounds of replication, you have some hybrids and some N14 only DNA strands. A second feature of DNA replication is that it is bidirectional. In this case, we see DNA double helices, and we see that helicase has broken the hydrogen bonds here to prepare for replication. When that happens, there's nothing fundamentally different about the right side and the left side. Chemically, they are the same. And what that means is that a DNA polymerase molecule could sit down in either of these directions on the fork and replicate. It is bidirectional, moving to the right and the left at the same time. As long as it's building 5' to 3', it doesn't matter which is right and which is left. A third thing that we know about DNA replication is that it is semi-discontinuous. And what this means is that some pieces of DNA are built in long continuous strands, but other pieces are built in short discontinuous strands. We know this from the Okazaki experiment done by a husband and wife pair, both of whom were named Okazaki. In this experiment, they had the ability to label DNA for very short pulses after replication started. You see in the green figure here that they looked at 20 seconds and 60 seconds after replication began. And then they could separate the resulting fragments based on size. So in normal healthy cells, when they looked after 20 seconds, they saw some medium-sized pieces of DNA and some very short pieces of DNA. When they looked after 60 seconds, that peak of short DNA was gone. And they hypothesized that that was because those short pieces had been glued together to create longer pieces. To test that hypothesis, they did the experiment again, but without the ligase, without the enzyme that does the gluing of small pieces to make long pieces. And when they did that, they saw that whether they looked at 10 seconds or 60 seconds, they were always seeing small fragments. Presumably this peak over here is the continuously replicated DNA. And those short pieces are the discontinuous pieces of DNA. And so we call those short discontinuous pieces Okazaki fragments. I'll come back to that in just a minute. The fourth thing that we know about DNA replication is that it is always built 5' to 3'. That's an inherent piece of the mechanism of nucleotide polymerization. The process of DNA replication requires several enzymes. In this picture, we see examples of each. So there's a helicase, which breaks hydrogen bonds using ATP. And what that does is separate the two parent strands so that they can be replicated. DNA polymerase cannot start from scratch. It can only extend from 3' hydroxyl groups that are already available from previously placed nucleotides. So in order to get started, we use a primase, which is an enzyme that creates a small RNA primer. There are also single-stranded binding proteins. These are necessary because if we used helicase to break the hydrogen bonds, but then helicase continued, what would happen is that those pieces of DNA would just reanneal, make their hydrogen bonds again, and go on their way. We need a mechanism to keep the DNA open long enough to be replicated, and these proteins bind to single strands and prevent reannealing. One of the other things that happens when you open the DNA helix is it super winds the helix out in front. If that were to continue, the DNA would become twisted so tightly that it might break and create double strand breaks. So we need a mechanism for relieving tension. Topoisomerase is the enzyme that does this. One of the ways it can do this is by actually creating a single strand break, allowing the DNA strands to unwind from each other and then ligating them back together before the polymerase comes in to replicate that section of DNA. So these 4 proteins are all necessary just for the setup of replication. DNA polymerase is the enzyme that will actually do the replicating. DNA is built in a 5' to 3' direction by a protein called DNA polymerase 3. This is your main DNA polymerase. And you see in the bottom that it has kind of a crab claw shape like RNA polymerase, such that DNA comes in, unwound, nucleotides enter. There's a guess and check mechanism. If the nucleotide that entered can create hydrogen bonds with the parent strand that's already there, there will be a condensation reaction and that new nucleotide will be added to the chain. If that nucleotide can't, hydrogen bond in that position, it will flow right back out again, and another nucleotide will flow in to try again. In addition, when these nucleotides are added, we cleave two phosphates off, and that nucleotide hydrolysis of those phosphates provides the energy to continue growing the chain. Remember that these nucleotides are ATP and GTP, they are TTP and CTP, and because these are deoxynucleotides, they do not have a hydroxyl group on their 2' position. So again, as we look at the bottom, what we see is that the chain itself that's growing is shown in red. It's growing from 5' to 3'. And the chain that it is reading is in orange, running from 3' to 5'. And new nucleotides will get added at the 3' end of the growing chain. One thing to consider, though, is that replication is occurring on both strands simultaneously. So often you'll see a picture that looks like this. And we are growing the chain in this direction. And maybe this is the continuous strand. So DNA polymerase is here and it just extends all the way out. And maybe this is the discontinuous strand. And so DNA polymerase is building this in small pieces. But with a picture like this, it's hard to know why that would be necessary. Why would you build that bottom strand in small pieces? The answer is that the two DNA polymerases and the helicase and the primase are all connected together in one giant complex. And DNA polymerases can only build 5' to 3', but the parents that are coming into this complex are anti-parallel, which means one of them is moving 3' to 5' to the left, while the other is moving 3' to 5' to the right. In general, that provides a kind of structural constraint to the whole system. And so what happens is the lagging strand actually loops around, and you see that over here. It loops around such that it is going in the same physical direction from 5' to 3'. As it was coming in, this piece of DNA was moving to the left as drawn. But when we loop it around, that same parent strand is now moving to the right in the same direction as the parent strand that's in anti-parallel being synthesized continuously. So this looping allows bidirectional replication to happen on both strands from 5' to 3'. But in doing that, on the lagging strand, you can only copy one loop at a time. And so the Okazaki fragments are actually the length of one loop. And then that loop will get dropped and a new loop will be created and the whole process will start again. There are several good YouTube videos about this, and I will point them to you on our learning management system. So here we are looking again at a 2D picture of how this works. And when you look at this picture, the Okazaki fragments don't really make any sense. If we imagine that this piece of DNA had been picked up and looped around so that it was going in the same physical direction, then we can see that each Okazaki fragment is one loop going around, being synthesized, and then the next piece of DNA would have to get picked up into a loop and synthesized next. Once we've done that, two things need to happen. First of all, the primers that we created were made of RNA. So those have to be removed and replaced. And that happens by an enzyme called DNA polymerase 1, which is kind of your fix-it polymerase. One of the things it fixes is the removal of RNA primers and the synthesis of DNA to go in. The other thing that we know has to happen is that each of these little segments between Okazaki fragments or where the primers have been replaced have to be glued together. And ligase is an enzyme that really just creates one phosphodiester bond between those fragments so that we have one continuous piece of DNA from 5' end to 3' end. The ends of the chromosomes are copied by an enzyme called telomerase. Because replication starts from a primer, the ends of a linear chromosome can't usually be replicated in the usual way. So if we were to look at this picture and start here, the bottom strand is being replicated continuously. The top strand is being replicated discontinuously through Okazaki fragments. At the end of this process, the RNA primers will need to be removed, and a polymerase would need to come in and copy that section. At the end of the chromosome, this is a problem because we know that DNA polymerase cannot start from scratch and can only extend previously created 3' hydroxyl groups. So in this position down here, there is no free 3' hydroxyl group. There's nothing for DNA polymerase to extend. If that was left just like it is, that region would never get copied, and chromosomes would get progressively shorter as we go along. This process is solved in some cells by an enzyme called telomerase. And telomerase works by extending the template strand, allowing new primers to be added for replication. So here is the region that would have been skipped. Telomerase carries its own RNA template, and it has polymerase function, so it can use that RNA template and extend the 3' end of the original parent strand. Once it's done that, a new primer can be placed, and DNA polymerase can copy this region, and ligase can glue these pieces together so that that piece of DNA doesn't get skipped. And in that way, the telomeres don't get shorter every time the DNA is replicated. So the mechanism of replication is constrained by the structure of DNA, by the fact that it's anti-parallel, by the fact that the base pairs are on the inside of the structure. And each part of the process of replication has been evolved to deal with a specific constraint. We'll look more at that in class.